Everybody, my name is Rna Sequencing. I'm from the University of Copenhagen and from the Metabolism Center. So since my master's study, which I also did in Copenhagen at the Institute of Preventive Medicine, people have been asking me with a seemingly very simple question. Why don't obese people or people who are living with obesity just lose some weight? And this turns out to be a question which is just really difficult to answer. And that's so I'll today talk about how we are addressing this question. And so throughout my talk, I'll be talking about single cell techniques that we are applying on mouse data. Now, we are also applying it on human data, and it could be data that's coming from the Danish National Biobank or from other research biobanks. And in fact, we're currently sequencing postmortem brain tissue, and also we're looking at liver samples and samples from muscle samples and adipose tissue samples from biobanks. However, this is very much ongoing. So all of the techniques that I'm explaining here, they can equally well be applied on this human data. It's just slightly more complex, but it works. Okay, so back to obesity. Currently, there is just one effective treatment against obesity, and this is a very new treatment, and that's the GLP-1 or the glucagon-like peptide-1 receptor agonist called semaglutide, which was developed by Novo Nordisk. And apart from, that's the only drug that's basically effectively reducing weights in individuals. So on the y-axis, you're seeing the change in weight, and on the x-axis, the time of this particular trial. So you can see that the semaglutide reduces weight of about 10% or more. And that's actually quite a lot, and that's good in obesity research. There have been many more drugs which all basically have failed to do this. So before this drug was approved by the FDA, which was earlier this year, basically the most effective option to treat obesity was bariatric surgery. So we surgerize an individual with obesity so that that individual would basically, the food in that person would bypass the stomach. And that's, it works, but it's just also having a lot of some negative side effects. And one of them being that there are many things you can't eat, and it's your life quality really is reduced afterwards. So this seems to work. The question is now how it works and can it be improved? What we know is that it seems to be acting through the brain. So there's a study here by people from the University of Michigan where they had a mouse model, and they removed the receptor, the GLP-1 receptor in brain neurons. And so in the control animal, you can see here, if you give liraglutide, which is just another name for semaglutide. So if you give the compound, you are reducing weight in the control mice. And however, once you're knocking out the GLP-1 receptor in certain neurons in the brain, you don't see that the liraglutide compound reduces weight. So we know it's acting somehow through the brain, but that's about it. We don't know how it acts through the brain. We know also that obesity has got something to do with the brain. So if you take results from genome-wide association studies and you look at these genetically associated variants, where are they expressed in the genome? Then you'll find that they are expressed at genes that are highly expressed in the brain. So here's a chart just showing you what we know about the genetics of obesity. So since 2007, there's been numerous studies identifying more and more genetic variants that associate with obesity. So now we know about 1,000 variants. So we know 1,000 sites in the genome where we know that this particular variant somehow impacts obesity. Now, in a couple of years ago, during my postdoc studies in the GIANT consortium, we looked at 209 different tissues and cell types, and we asked a quite simple question. These genetic variants I just showed you, where are they expressed? And are they expressed in certain particular tissues throughout the body? And what we found was quite strikingly, namely that all these variants, they seem to be located in parts of the genome that is expressing genes expressed in the brain. So we know now that common obesity has got something to do with how our brain works. So it's not surprising us that semaglutide or liraglutide also seems to work through the brain. Okay, so we set up a study, and that was a PhD student in my group, where we first asked if we take certain brain areas and look at gene expression changes, in which parts of the brain are we seeing the strongest changes induced by semaglutide, right? And right, so here we are using bulk RNA sequencing, similar to what Victor, I think, just told you about, and we look at what happens in any of these six brain areas after treatment, and we saw that the most treatments occur in this lower part of the brain stem, so in the hindbrain, and it's in this area postrema nucleus. So we now knew, okay, there are a lot of changes induced by the drugs, this drug here, and it's probably going through the area postrema, which is an interesting area because it's at a part of the brain where there's no blood-brain barrier. It's a penetrated blood-brain barrier, so that the blood-borne signals, they can signal to this area. It's sort of this area can react on compounds in the blood. And we obviously, obesity isn't just a brain disease. It has got something to do with the body and its communication with our control center up here. So we knew then, okay, probably this drug, at least from this work here, from this experiment, the drug is inducing a lot of changes here. So here it's where we then focus our single cell analyses, because these are still analyses that are quite expensive to do, and we couldn't single cell sequence all of these areas, which technically we could, but it would have been very expensive. Illumina would have been happy. There's more Jacobs here, but there's no way you can do it funding wise. So we had to focus on a certain area, which was area postrema. Then we applied two techniques. One is single cell or single nucleus RNA sequencing, which is relatively straightforward. You take the tissue of interest, and totally same applies for human tissue. It's frozen material, so it could perfectly well come from a biobank, just needs to be SNP frozen material. We mechanistically dissociate nuclei, so we only work on RNAs from nuclei so that we can work on frozen tissues. And then once we have these nuclei dissociated, we typically do a FACS sort here just to remove some embryon RNAs that are within a nucleus. And then we do RNAs. We barcode each cell, and then we do RNA sequencing. So for each RNA, we know which cell a given RNA came from. All the RNAs here are having a red barcode. So we know all the red reads in our data set are coming from this particular cell. So this way, we sort of have a single cell data set of gene expression data, gene expression changes in this area. We also did something called single cell ATAC-seq, and this is a technique that allows us to profile open or closed chromatin. So as you know, our DNA in every cell is wrapped around nucleosomes, and only the DNA in open areas, so areas where the DNA is not wrapped around a nucleosome, can be transcribed, right? In these open areas, you can have a transcription factor, set of transcription factors that are recognizing some DNA motifs. They can recruit the RNA polymerase, which then can drive the expression of a gene. So we are interested also in open chromatin because this will help us to better understand how genetic variants impact obesity, and I can get back to this later. We did both of this, not in the same cell, but in separate cells. There are now also protocols from 10x Genomics where you can measure open chromatin and RNA expression levels in the same cell. That might be interesting to some of you. That's possible now, but we are measuring how accessible is some DNA in a given cell and how much RNA is expressed of a given gene in a given cell. We did it for this area postrema, nucleus, and we got some distribution of cells. So why did we do this? Well, we knew there were a lot of changes in that area, but we didn't know whether the semaglutide, whether that drug induced changes in neurons or in some glial cells on oligodendrocytes. So we wanted to catalog what are the cells that are present in that area. And because that catalog didn't exist, we did it in these animals. Now, as I mentioned, we have human samples, so we are about to construct a similar catalog for humans here for this area. So typically what we do is we look at all cells and isolate and find bioinformatically and take out all neurons, and then look at whether they're expressing certain genes that are, so what are the genes that are particular to the biological cells in this area, what genes are expressed there. And doing this, we can then give them names. So some of that's just a note here. Some of them are expressing glutamatergic cells, so they are expressing a glutamatergic neural transmitter. We call them GLUT1 to 20. Some of them are expressing other neurotransmitters. So we give them names now. And what we then can do is to also figure out what receptors are they expressing, because we're interested in which of them are expressing the GLP-1 receptor. And from the literature, we knew the GLP-1 receptor is expressed there, but we nor any other group knew where is the GLP-1 receptor expressed in this area, and where are other relevant weight module regulatory receptors expressed here. We and others didn't know, and this is one of the questions you can ask into such a single cell data set. So we found that the GLP-1 receptor was expressed in this GLUT4 population, and since then we have also found that if we sequence more cells here, these are actually three populations. So the GLP-1 receptor is there, and it's expressed in these three populations. Then we also found that the GLP-1 receptor is expressed in the NTS, the nucleus of the solitary tract, which are cells that are coming from a neighboring area. And they could be interesting as well. We also found that there are two other interesting genes that are expressed over here. One of them is calcitonin, and the other one is amylin. The other one is RAM3, sorry. And the calcitonin receptor and the RAM3 receptor, when they co-localize, they can sense amylin. And amylin is another, it's a peptide that's secreted by the pancreas, which is also weight regulatory. And Novo Nordisk and Eli Lilly and others have been working for more than 10 years now on synthesizing a receptor agonist for amylin. And I think they are about now to have a compound they're working with. It's called cagrilintide from Novo Nordisk, and it's an analog that's targeting this receptor complex over there. So we saw that this receptor complex, it seems to be expressed here. And it's interesting because we know the GLP-1 receptor is expressed here, and also from some ongoing trials, we know if you do a dual agonist where you fuse semaglutide and cagrilintide, then you are seeing a additive or maybe synergistic effect on weight loss. You're seeing much higher weight loss. And it's probably because they are acting, so semaglutide is acting here and cagrilintide is acting here, and they are not engaging the exact same pathway, so they must be somehow complementing each other, these two cell types here, because otherwise, if they did the same, then you wouldn't see any additive effect of this new dual agonist treatment. So we're quite interested in these cell types here. I just wanted to show you one slide on how the open chromatin data looks. So what I showed you was the single cell data. Now, if you look at open chromatin and you just say, now I'm clustering all my cells based on how their open chromatin landscape is, then this cluster plot looks very similar to the previous one I showed you. We are basically finding the same cell types, and they group in similar ways. Here we have GLUT10, for instance, the one who expressed the amylin sensing receptor complex. So this basically told us that if you look at open chromatin and RNA expression, it's quite similar. They're not exactly doing the same, but some RNA, in order to see expression, you need open chromatin, so they correlate these two data sets. And I can get back to how we are utilizing this data. I just want to show you how the chromatin data looks in this brain area. So the last analysis we did was to say, well, now we know all of those cell types. So what if we now repeated the analysis I showed you in the beginning, where we ask whether if you have all the genetically associated BMI loci, and those are pointing us towards 1,000 places in the genome. Now, if we look at these 1,000 places and we check the genes that are expressed in these areas, and we ask, are these genes expressed in a certain cell type, not in a certain organ or brain area, but we're now checking whether these 1,000 genes are expressed in a specific cell population. So we're increasing the resolution on the figure I showed you in the beginning. So I'll show you the results in a plot that looks like this. So here we have all our cell populations. Here you can see whether the population is from the area postrema, the nucleus of the solitary tract, or part of the vagus nerve, so the AP, MTS, or EMB. And on the y-axis, the higher a given dot, the more significant that cell type enriches for genes or expresses genes that are also co-localizing with genetic association studies for obesity. So what we found was that we have 4 cell populations which are expressing genes that are also within genetically associated loci. And these are the amylin sensing neurons, the CALTS-R-RAM3 receptor complex neurons, the GLP-1 and 2 GLP-1 receptor neurons, and one population which we don't understand anything about, nor is it described in literature. So that's one more interesting population which we are currently working on figuring out what it does. We don't know yet, but we're curious to find out. But the other 3 make perfectly sense. So right. So, and this is sort of an important point. So if you think about it, we came in from a mouse study, profiled all the cell populations in the area postrema, and then looked at whether genes that are expressed in these cell populations are also coming up in genetic association studies for obesity. And when doing this, we find these 4 populations. And then 2 of them are likely drug targets for semaglutide, and 1 is a likely drug target for cagrilintide. And these are the 2 most promising drugs that are currently being developed for obesity. So it just shows you so I guess we were lucky, and also we knew where to look because we knew that SEMA is acting on this area. But this shows that this idea where you take single gene expression data and integrate it with genetic association data is a powerful approach to sort of identify some interesting biology, which then obviously we need to do a lot of additional studies to figure out how they impact obesity. And so we redid the same analysis I showed you for the chromatin accessibility data. And I'll just leave out details here and just tell you that if we integrate the chromatin accessibility derived cell types with our genetic association data, we basically find the same, more or less. And so just the last experiment we did with a close collaborator was to have build a DREAD model, slightly more or less like you see it here, where we have a transgenic animal where in that animal, under the promoter for CALTS-R-RAM3 are expressing a receptor that can activate with CNO. So in this mouse, when we give CNO through the drinking water to this transgenic rat, then we can activate this CALTS-R-RAM3 population through this DREAD setup. So here we then, once we had built the mouse, we asked whether if we now activate this population, the CALTS-R-RAM3, what happens? And our prediction was it reduces food intake because amylin is reducing food intake. And that's exactly so what we saw. So on the y-axis, you see cumulative food intake, on the x-axis hours. So in the blue group, that's the group where we activate these cells. In the red, this is the group group where we activate the cells, the magenta, the big one. So and here we see that they eat less. So this basically, in the red, proves that this cell population is modulated weight. To conclude, GLP-1 and amylin sensing cells are located in the area postrema. We can see that the GLP-1 amylin sensing cells enrich for genes that are implicated also in human obesity. And so we were able to do these discoveries using single cell techniques and genome-wide association data, and to basically sort of fast track some of the discoveries that have previously been made for obesity. And I think what hopefully you can basically take home from it is that single cell studies are feasible also in humans now, and it's a powerful way to sort of agnostically figure out what cell populations are present in a given sample, which cell populations are responding. And if you want, you can also integrate it with genetic association data, because that may give you an idea about how these cell populations relate to different traits and diseases and phenotypes. And so with this, I wanted to acknowledge Mette, who has been doing most of the work I was showing you, and Martin, who has been setting up the DREAD experiment, and then a number of people who have been helping us to do the in vivo, to do the semaglutide in vivo studies for us. And thank you for your attention.